Hello, welcome to El Zoominario. Today we are very excited to welcome Hector Baños Cervantes. He's going to be talking about mathematics and DNA. Hector is from Mexico, born and raised. He moved from Mexico to the US in 2014 to do his PhD at the University of Alaska, Fairbanks. Currently, he's an assistant professor at California State University, San Bernardino. His main research is on mathematical phylogenetics. Thank you so much, Hector.
Yeah, no, thank you for inviting me. It's a pleasure being here. So what I'm going to be talking about is pretty informal. It's about some of DNA applications and how math come into play. Some of these things do not work directly. So some of my friends and colleagues work on these type of projects, but I just want to share to you a little bit of an overview of what is people doing. So first, let me start with DNA and just something else. I'm going to be super informal on the biology and the math. So just as a heads up, so just to have everybody on board. So DNA is the molecule that contains the genetic information of living things. Really, broadly, you can think about a DNA as a recipe. DNA is stored as a code of 4 nucleotide bases, A, G, C, and T. And in DNA, it's a double helix a structure where A always binds with a T and G always binds with a C and vice versa. And those form base pairs. So therefore, if you only know one half or half of the DNA, you can always get the complement by looking at what do they should bind. So for us, mathematically, DNA just a sequence on a 4-letter alphabet, A, G, C, and T. Now, one thing about storage information or data is the following. So if you think about a hard drive or a hard disk or a flash memory, one of the biggest issues they have is the data retention, which it's less than 10 years. This implies that after 10 years, you have to reread the information on that hard drive or transfer it to another or make sure that information is there safely, because they cannot guarantee that information will be there after 10 years because it degrades. And that becomes a big problem, for example, for the US government, because they have to store tons of documents regarding a lot of bills or police reports, and a lot of things have to be in file. Maybe they will never use them. They have to have them stored just in case. And having to reread the document every 10 years and with the amount of data they have could get completely burdensome. So one solution was to use bacterial DNA. And how does this work is as the follow. So suppose you have a file. It could be a song, a video, whatever. That's the file that you have in your computer. That file on your computer is just a binary sequence of 1 and 0s. It's not hard to convert those 1s and 0s to a 4-letter alphabet of A, G, C's, and T's. I'm sure if I give you enough time, you will be able to come up with a way to get a map that gets that sequence to that sequence. Then a machine can synthetically build a DNA encoding that a sequence that has the nucleotides, and then you can store that DNA sequence. And if you get, by a rare chance, you need to get that file, what you can do is sequence that DNA and then apply the inverse map to get your original binary sequence and get it to recover your file. One advantage of this is that bacterial DNA takes a long time to degrade, around more or less 100 years that your data will be safely stored. Another thing that is positive is that hard disk or flash memories, they get outdated with time. So if you remember this type of USB, this is USB-A. Nowadays, as time passes, more computers just have the new USB-C. So therefore, you have to change the technology. With the bacterial DNA, you always have to sequence it because we will keep to doing that. Now, this, of course, is for files that you will rarely use, like the ones from the US government that I'm saying. So people are looking into that. Now, one of the problems is that sequencing data sometimes could be problematic, as mistakes can be made when you have the same letter repeated multiple times. And therefore, it can tell you that there's less letters or more letters than there actually are. And that, if you give a wrong binary sequence, could result in a completely lost file. Or maybe not, maybe you didn't even notice there was a mistake. So the problem, mathematical problem here, it was ways to find a map that will be adequately robust, better say, to these changes. So the things used here is a lot of algebra and tensor theory, and they use all that to guarantee and minimize the error rate and a failure of that. And I know that Microsoft have already developed and implemented this technology, but they just want to use it for larger files. And therefore, there's a lot of mathematical tools needed. Another applications I've seen is about a case of a man. The case was called the puppy dog. It was a person that was unfortunately treating really bad his dog. And this person, they found the dog and therefore had access to their DNA. And and they found DNA from a dog in the kitchen of this person. Because this person was claiming the dog that they found, it was not theirs, they had to really match the sample found in the kitchen and the one of the dog. Now, I think this, to my understanding, this was the first time that canine DNA was used in Massachusetts for a trial, like as evidence. So therefore, they had to bring Dr. John Rhodes, who's a collaborator of mine and PhD advisor, to give his assistance in a statistical analysis of the DNA evidence. So they have to provide this analysis saying like the probability of the two samples not matching, it's 1 in billions, and therefore they should be the same dog. And because this is a trial, they have to be really sure it was the case. And that brings us to another related case, which is the case of Kimberly Bergalis. Kimberly was a patient who allegedly was infected with HIV by her dentist during a tooth extraction. So the problem is, how do they guarantee that he was infected by the dentist and not it was something else? So here, and this is more or less more from what I work with. So what they did is they took 10 people around the area who also were infected with HIV, and the CDC, the Center for Disease Control, performed a phylogenetic analysis that I'm going to talk about that later, what it is, of the DNA sequence of the viral envelope genome. What does that mean? For each of the 10 patients, they look at the viral envelope genome, and then they look at which ones are the most closely related, or which ones are really alike. And when they obtain something of this sort, where of the 10 people, 6, they found that the virus was really closely related to that of the dentist. And this case, it's a little bit polemic. There's a lot of polemic into it. It has to do with politics and a lot of things, but I think it was something in terms of bringing awareness and this kind of analysis. And this same analysis can be done not just for HIV, but for other viruses. So the same techniques can be applied. And this exactly was done with coronavirus. So the reason we believe coronavirus came from the bat, or it's close, the most closely related virus is from a bat, is because they realized a similar phylogenetic analysis and found that the most closely thing was the bad coronavirus. So let me talk a little bit more about this. So in order, the fundamental thing here is evolution. The fundamental term here is evolution. And evolution says that living things do not appear out of nowhere. They have an origin and change over time. Darwin was one of the first ones to propose that species were related and have a common ancestry. And in his notes, he wrote the word "I think," and then a tree relating different species. And it's really common, the finches example, but if you want, you can Google. So loosely speaking, what we have here is a tree of life showing all the evolutionary relationship between different species. There's an origin of life where every living being evolved from. And as time passed, new species were appearing by speciation events, and some others were extinct, like the dinosaurs, that maybe we don't see right now, but at some point they happen. So why do we believe in all this? Is because there's a lot of, in DNA, we can find the tracings of this. For example, we share more than 50% of our DNA with a banana. So what they do in this phylogenetic analysis is look how closely related the species are based on their DNA sequences. Now, there's a lot of mathematics involved in these processes. The first one is alignments. So when we talk about comparing different DNA for species or genes, we have to have in mind that the nuclear genome or the DNA between species, it's really variable. So humans or Homo sapiens, the number of nucleotides or length of the DNA is 3.2 billions, where Drosophila, which is the fruit fly, has 170 millions, and Physicomitrella patens, which is a kind of moss, has 408 millions. So to compare genes properly, they have to be aligned. And for that is, you want to find the sites in DNA where you can compare those. And that involves a lot of algorithms that, in this particular case, a function that will be punishing some of the different ways you can align the sequences. And you want to find the best one where things are more alike. For that, there's a lot of computer science and biology and mathematics involved to find good, and alignment algorithms are evolving. As time passes, they're getting better and better and better. Once you have a set of aligned sequences, what you want to find is the phylogenetic tree, the ones that show how the species evolve. For example, here we have the one relating orangutan, gorilla, chimp, bonobo, and human. And here we can see that the most closely related are chimp and bonobo, and they have an extinct common ancestor here. And then the next one closely related is human, then gorilla, and then orangutan. In these processes, there's a lot of mathematics involved, a lot of computer science, statistics, and of course, a lot of biology, because we want to develop models that describe these processes as accurate as possible, but that they are manageable. The thing that I'm most interested in is hybridization, and that occurs when two different species merge genetically to create a new one. And here I have a mock-up example, like Kermit the Frog and Miss Piggy having a pink frog, but this is known to happen a lot for some type of fish, plants, frogs, salamanders, among others. And in this case, we don't use a phylogenetic tree, but a phylogenetic network. Here, in order to properly model how hybridization and how things occur, we need to look at other processes that happen. And one that is common is gene tree incongruence. So when you look at different genes, which are kind of the building blocks or the instructions for in DNA, there are different genes that show that have different evolutionary histories. So here I have an example of butterflies where you can see in blue a lot of gene trees showing that these two are more closely related, but other genes, these two are more closely related. That is called gene tree incongruence. And that could be due to different processes like hybridization, incomplete DNA sorting, loss of genes, among other processes. I'm not gonna elaborate much on these ones, but I want just to show that there's a lot of biological processes happen that we need to account for in order to get accurate estimates of the evolutionary relationships we want to recover. Without going through much detail, I'm really interested in the network multi-species coalescence model, which is a model that describes how genes are formed within species networks in the presence of ILS and hybridization. This is a stochastic model that is modeled like using Poisson processes. Again, I'm not going to go through the details, but it involves a lot of stochasticity, and we want to recover the information using these models. So the main question we want to solve is, given a DNA sequence or DNA sequences sampled from evolution models, what properties can we identify and we recover from the network? So given a DNA sequence, what can you identify from the network? And that is what I work on mostly. And again, this is pretty informal, but that is more or less what I do. And we apply a lot of mathematical models. And specifically, and this is just what I'm currently working at, is I'm working with Kelsey Williamson from, she's a PhD student at Dalhousie University, along with Andrew Roger and Ed Susko. And one of our projects is develop a new phylogenetic data set for pinpointing the root of a characteristic tree. This project involves trying to use an outgroup to find where the most recent common ancestor for all eukaryotes is. Now, when modeling these types of problems, we're talking about evolutionary relationships that occur like billions of years ago. So one has to be careful in which techniques are being used and what models. I'm also involved in a project called GTR linkage changeabilities, where, without going through much details, we want to improve current methods to estimate phylogenetic relationships for these evolutionary relationships that occurred billions of years ago. Another one, I'm working on networks, the things I just talked about, and along with Jonathan Mitchell and Elizabeth Allman and John Rhodes, we're trying to show how what properties of the species networks are identifiable, and also with Elizabeth Allman, John Rhodes, and Cecile Anne and Jianqing Xu, we're working on other type of identifiability problems. Some of these ones are very applied. That is, we want to develop algorithms that biologists will be able to use to recover meaningful relationships. And I think that is for me with respect to the research. Again, pretty informal, but more or less to give everybody an idea of what I do. And about when I was asked to talk about something regarding my culture, I want to just tell you about a legend that I love, which is the legend of Popocatépetl and its Tzatzíhuatl, which is a really beautiful love story. So I'm gonna read it because I don't want to butcher this, but let me give you a context. Popocatépetl, it's a volcano in Mexico, and Tzatzíhuatl is another volcano that is really close to. So the legend goes like this. In ancient times, when the Aztec Empire extended its dominion over the valley of Mexico, neighboring towns were subject to heavy taxation. The chief of the Tlaxcaltecas, long-standing adversaries of the Aztecs, could no longer bear this oppressive burden and resolved to fight for the freedom of his people. Among the chief's children was incomparable Princess Iztaccíhuatl, renowned as the fairest of all princesses. She had fallen deeply in love with Popocatépetl, a valiant warrior from her father's tribe, renowned for his striking handsomeness. The two young souls professed an unwavering love for one another, and before departing for battle, Popocatépetl approached the chief, seeking his daughter's hand in marriage. The chief gladly consented and made a promise to celebrate the union with great joy upon Popocatépetl triumphant return from the war. Filled with hope and determination, the brave warrior made all the necessary preparations and embarked on his journey, carrying with his heart the assurance that the princess would eagerly await for the reunion to consummate their love. However, a jealous rival of Popocatépetl, envious of the profound bond shared by the young lovers, deceitfully informed the princess Iztaccíhuatl that her beloved had perished in combat. Crushed by the weight of grief and consumed by sorrow, the princess succumbed to her despair and passed away, unaware of the falsehood that had shattered her spirit. Meanwhile, Popocatépetl emerged victorious from the battle, journeying to find his beloved princess. Yet, upon his return, he received the devastating news that Iztaccíhuatl untimely demise. Overwhelmed by anguish, he roamed the town aimlessly, haunted by his loss for countless days and nights. Determined to honor their love and ensure that Princess Iztaccíhuatl will never be forgotten, Popocatépetl commanded the construction of an immense tomb beneath the sun. 10 hills were piled together to form a colossal mountain, symbolizing his eternal devotion. Gently cradling the lifeless body of his princess in his arms, Popocatépetl ascended to the mountain summit and tenderly laid her to rest. With a loving kiss upon her cold lips, he took up a smoking torch and knelt before his beloved, forever guarding her eternal slumber. From that moment onward, they will remain together forever, facing one another. As time passed, the snow cloaked their bodies, transforming them into two colossal volcanoes, inseparable until the end of days. The legend continues that whenever Popocatépetl, this volcano, the warrior recalls his beloved, his heart, which preserves the fiery embers of eternal passion, trembles, causing his torch to emit smoke. Thus, to this day, the Popocatépetl volcano continues to release fumaroles. So here you can see these are the two volcanoes, and the story is, that Iztaccíhuatl, you can see the shape of this volcano is the shape of a woman laying down, and the shape of Popocatépetl is the shape of the Tlaxcalteca warrior kneeling down. And every time there's smoke from them, one of them is resembling their love story, or that they are still caring for each other. And I think that's it for me. I'm happy to take any questions.